engineering program. And we are all students. Oh, I'm very happy to have you all here. And we will have a lecture from Professor Hiroyuki Oda. He is a president of Ibadan University, Japan. So I think this is a good chance in this occasion. We will have a lesson, study lesson from him. So hopefully we can get sight how to deal with climate change from the microbiology point of view. Yeah, so please pay attention. Please take notes during this lesson. And here, without further ado, please welcome Professor Hiroyuki to come to give a lecture. Please give a broadly.
Okay. I keep. Okay, can you hear me? No. どれだろう?
You want also use, please use this.
YouTube.
Yes.
Okay. Thank you for the introduction of me. So thank you, Anissa. Just before this lecture, I talked with the dean, and we had a discussion about the agriculture and technology. So the combination of the agriculture technology is very important, mainly for the sustainable future. So my research field is the soil microbiology, a little different from your study, the environmental engineering. But I hope you will get something about my lecture. So I start. So my lecture start with this message. 3 years ago, about 30 scientists that made a message to the world. The title is the Science Warning to the Humanity, Microorganisms and Climate Change. So essence is here to understand how humans and other life forms on Earth can withstand anthropogenic climate change. It is vital to incorporate knowledge of the microbial unseen majority. That is one point. And another point is the impact of climate change will depend on the responses of microorganisms, which are essential for achieving an environmental sustainable future. So, and next slide. So this is a summary and the aim of this lecture. First half is the carbon cycling. We think carbon cycling, terrestrial carbon cycling, thinking behavior of fresh, labile, and old recalcitrant carbon. How do we reduce the use of old carbon, fossil fuel? How do we decrease the CO2 emission from fresh carbon? Do you know new about fresh and old carbon? So now I hope you understand the difference between the old and fresh carbon. And the second half of my talk is about nitrogen recycling, the complex nitrogen metabolism. So I have a question to you. How do we make soil management practice able to reduce emission of nitrous oxide from soil? Okay. This picture shows the geochemical cycling of element. Center is drawn the CHO cycle. now you know photosynthesis is the solar energy is used for the splitting the hydrogen and oxygen, generate the hydrogen atom combined with CO2 and result in the formation of CH2O. That's the organic carbon. This is photosynthesis. The other part is the respiration. Animals and also including the plant, microbes, degrade organic carbon and liberate CO2 and hydrogen atom combined with oxygen and making H2O, water. This process, the biochemical energy produced. So CHO cycle means energy conversion system from energy to biochemical energy. So this is the fundamental energy conversion system on the Earth. Cycling is a nitrogen and sulfur. In the case of nitrogen cycle, the member is nitrate, nitrite, ammonium or ammonia, and organic nitrogen and nitrogen. So the cycle here, it's a nitrification, it's a ammonia oxidation, nitrite oxidation, resulting in the nitrate formation. In anaerobic environment, nitrate converts to ammonium or nitrogen. So intermediate is NO2 nitrate. This anaerobic process is supported by the hydrogen liberated from the degradation of organic carbon. So second half anaerobic part is required for the anaerobic organic carbon derived hydrogen. That's the same for sulfur cycles. Elemental sulfur, sulfate, hydrogen sulfide, the same story. So, on the Earth, we have three major CHO cycle, nitrogen, and so one point is the CHO cycle is the energy system in Earth ecosystem. Second, nitrogen and cycling requires the interface of aerobic and anaerobic cycle. Third, organic carbon, in this case, carbohydrates required in the anaerobic nitrogen and sulfur conversion. So this is the fundamental things for understanding geochemical cycling. Is that okay? It's the quite fundamentals. Oh.
Yeah.
Yeah. Okay. Done. Yeah. Okay. Firstly, I talked about old and new carbon. So now I explained about the CHO cycle like this. And it's a fresh, I termed this organic carbon is a fresh carbon because of the, now the plant photosynthesis is making the fresh carbon. And we use the carbohydrate as energy source and liberated the CO2 and also the H2O. Prehistoric time in the past, CO2 converted to fossil fuel, old carbon, we term old carbon. First photosynthesis made old carbon. Now, at presently, we use old carbon as energy source. So, extra CO2, that is the result in the global warming and climate change. So, if this cycle is the reaction is equal, no increase in the CO2, but we use old carbon. That results in the extra CO2. That is mean to the global warming. Here shows the yeah, of the and fresh carbon cycles. Here the colored part is the supported by the microbes. The blue color is the human activity. Yes. So atmospheric CO2, the amount is 700 up to 800 petagram. The photosynthesis, the organic carbon, this cycle, and microbial decomposition result in the CO2 formation. That's it. The cycle explain extra CO2. So, 2001, the ratio of microbial activity to activity by man is like twofold higher than microbial activity. Do you think that is a good balance or much much bigger than the whole microbial decomposition? But this fraction very small, but the effect on the climate change very important. Then I get the EPA from this sphere. Summarize the content of GHG. So the carbon dioxide account for 65%, mainly as a fossil fuel and the industrial process. This is only to the from old carbon. Also, the carbon dioxide liberated from the soil, forestry, and use 11%. That is mainly the fresh carbon. We have another GHG gas, the methane and the nitrous oxide. Compare in relative to the carbon dioxide, warming potential is the higher than for the methane to 23 times, and the nitrous oxide about 300-fold higher than carbon dioxide. So question, how do we reduce the use of old carbon, fossil fuel, and another here, how do we decrease CO2 emission from fresh carbon? So we need to think about new and old carbon. And also important is the nitrous oxide. It's a much higher warming potential, about 300. So how do we make soil management practice able to reduce the emission and flow from soil? So here I will introduce some results from the management practice. Yeah. This is the summary. We think future of sustainable society, and historic time, the past, and now, and the future. We are dropped the already fossil fuel use, the level of CO2, but it is a problem So that we developed the biofuels with the present photosynthesis, immediate use CO2 use. It's the carbon neutral cycle. And we don't use biomass. Directly solar energy change to electricity, photovoltaic module. So this is decarbonization. So we have two options. The carbon neutral way, depending on the present photosynthesis. The other is independent of CO2 cycle, directly change to solar energy to the electricity. So it's the future. But already the solar panel covering is increased, so the we much use for solar panel in place of the fossil fuels. That is the general tendency. But still, we think further. So I introduce our program. Ibaraki University challenged to DAC, direct air capture of CO2 with a recovery. We use the synthesis on use of materials. So we propose new carbon cycles depending on the atmospheric CO2, and that means making dependent system use of atmospheric CO2, synthesis of methane from CO2, and use of hydrogen. This is the one point. And the other point is neutral, carbon neutral. This cycle is independent of normal natural carbon cycling, just artificial cycling, recovery, synthesis use. This we propose. And one of the several researchers in our university produced the new material directly changing CO2 to the such as methanol. So no used as a fuel. So this is our propose another way of carbon independent system. It's engineering. So return to the agriculture. I show the several data. CO2 emission from soil that is increased. This data is literally old, the 2005, published in Nature, carbon losses from all soil across England and Wales, 1978 to 2003, 25 years difference. They measured just only the soil carbon content. Left of the panel shows the original organic carbon content. Dark colored spot means the higher original organic carbon. Pale spots stand for the lower organic carbon. 25 rate measured again, same spots, and the difference during 25 years. So this blue colored spot that higher than other spot. So per year, per kilo, minus 2 gram decrease. So higher organic carbon site correlate to the large loss of soil carbon. That means soil carbon decreases annually. They made an equation collecting data, and this data is rotation graph is the human use, and the permanent graph is a non-agricultural soil, and combined all. They made a quite a good correlation with the line. The rate of change in this organic carbon is 0.5 minus 0.0187 times original organic carbon. That means that annually CO2 emitted and the rate of loss increase organic carbon was more than 2% per year in solid organic carbon, higher than 100 gram kilo soil per gram. So this is the fresh carbon loss from soil. Another researcher measured directly soil respiration. It's published in 2010. They estimate the annual global soil respiration rate and measured the value of the 98 petagram carbon. If agricultural area 85 petagram carbon, and the increase rate is 0.1 petagram carbon per year, it's quite big. And increase in the soil respiration means loss of soil carbon, or another story they also present. Anyway, for us, from soil CO carbon converted atmospheric CO2, that is the evidence. They analyze further grid price with the A and the horizontal axis, solution at the proportion percent. B temperature cell, temperature zones, and C shows the tropical cell. Of course, tropical region showed higher respiration rate comparing with the high latitude cells. The point is here, how the largest absolute change over this time period occurs in tropical grass regions. Respiration, soil respiration in high latitude ecosystem has the largest rate of change, consistent with the large carbon stock in greater degree climate change being experienced by these areas. So high latitude area, the emission of CO2 from soil is more evident. Yes. Thank you. So the discussion occurs about such data. So example here, the comment Schultz and Freibauer, consistent on SOC occurred independently with soil properties, challenging our knowledge about soil organic carbon stability. The result SOC in carbon-rich soil contains higher fraction of stable carbon than does that in carbon. Yes, losses occurred in soil all land use conditions, and there is no apparent single factor other than climate change that could degrade non-agricultural soil. The observation is contrary to the view that soils ultimate large-scale sink for carbon. Yes, soil carbon is no longer a sink of carbon. But we think further. Now I talk that the fresh carbon, but the fresh carbon is not uniform, ranging from level to calcium. Organic carbon composes various form is easily decomposable, the microwave carbon. The humidified carbon is less much resistant to the decomposition. Inert is carbon is almost inactive. And the geological process converted inert carbon to fossil fuel. It's a very it depends on much time. Generally, the microwave carbon contain high nitrogen compared to the inert carbon. Also, the microcarbon is contain lower lignin compound relative to inert C. So we think the decomposability of these carbon fractions. So it's not the solid simple. We think also think about the carbon fraction for the fresh carbon stability. So, yes. Also, the nature, the 2010, the increase in the soil registration is either loss of recurrent carbon or enhancement of fresh carbon cycling. It proposed two assumptions. One assumption is that the increase in the soil respiration means old carbon decomposition. So increase the atmospheric CO2. Yeah, this assumption. The other is the global warming enhance the photosynthesis, also the respiration of the plant and soil microbe. So increased soil respiration means supported by higher photosynthesis rate. In that soil carbon loss does not occur. Does not occur. So we have to function now in this the what is occur in the soil, fresh carbon decomposition, old carbon decomposition, or just global warming influence positively to the photosynthesis and the soil respiration. Yes. I move to further details, so I hope you follow me. Historically, we think soil contains uniform carbon. And that panel A, mean annual temperature and turnover time. Turnover time is the decomposition rate. The turnover time is low. That means the rate is high. So the lower turnover time, the higher decomposition rate. If we assume one uniform soil fraction, that is independent temperature increase. But we divided three fractions. First, decomposed carbon pool, and the intermediate and very slow. It's a labile carbon and recalcitrant carbon. In that case, response of two pools are different against the temperature rise. Very important line here. Very slow pools more sensitive to temperature increase. That's the point. Global warming may importantly affect the composition of the recalcitrant, very slow carbon pool. Why? Yes, Davis and Jensen proposed that this is a quite beautiful response of soil organic carbon, intrinsic temperature sensitivity. That means that the simple substrate and complex substrate response of these carbon against the temperature rise is the complex carbon required more energy for decomposition. So increase temperature that support the decomposed activity of, now, of course, microbial activity. So increase like this. So another factor is environmental constraint. That is the carbon compound solved or dissolved inside the aggregate or outside aggregate, or aerobic, anaerobic. Water stress is rich water or poor water, frozen or unfrozen. So if carbon compound presented inside the aggregate, that is a protection from against microbe. So the stable state. So that we see two ways. That is the molecular structure of carbon, and the other is the environmental factors like this. So with an aggregate and aerobic, the water content and the temperature. Are you frozen or not frozen? Yes. So now I talk about the microbes. Please imagine that the soil where you pick up soil from ground and touch like this. Good soil is you can feed aggregate. Bad soil is no aggregation. So if aggregate present like this, right? Small aggregate get bind together, making and the large aggregate. So inside the aggregate, if organic matter present inside the aggregate escape from microbial attack like this. That is the aggregate protection. If the soil contains only a large particle, organic compound do not enter the inside. So the easily microbes can eat outside organic compound. So aggregation is very important. Here, this is a hunger hyphae. Hyphae is mainly used outside aggregate. And bacterial cell is smaller than fungal cells. The bacterial cell can enter, but they can't enter the inside. So that's the aggregate protection. Okay. I like one experiment, very simple experiment. Just collect the soil and made two portions. One portion is break aggregate. The other is intact. And put in the beaker and our flask, Maya flask, and made a stopper and the incubation. That's all. But periodically, air sample and measure the CO2. So that it's a structured aggregate and crushed aggregate. So how about the result? Yes. Simple experiment showed quite a dramatic result. In this case, the two types of soils, the NT, no tillage soils, no tillage. The other is the conventional tillage. Disturb the soil, no disturb soil, NT. And intact aggregate, crushed aggregate from no tillage soil, crushed aggregate shows this CO2 emission. Intact one is lower. Then means aggregate protect organic carbon. What that means, crushed aggregate microbe can eat them, but not crushed one microbe cannot eat them. So in soil, the aggregate formation is very important for CO2 emission from soil. Yeah, the data, empty, no tillage, and the conventional tillage, the relativity, protect, yeah, like this. But tillage treatment is very important for the carbon stability in soil. It's not the same result, but I showed that the particle side here. We all this experiment that no this time just that make the line for the seed for seedling. Yes, the other is the this time the soil by machine. I later I say that soil, and you can imagine that it's a aggregate, but we can divide side of aggregate by seeds like this. Yes. So the same result we obtained, also that conventional to tillage to no tillage increase the carbon content, and the aggregate carbohydrate also changed. No tillage result is much higher carbon content, the large particle, and getting small in the this particle side. Also, the annual carbon balance here, one collaborator, they no tillage carbon no tillage and conventional tillage. Annually, the tillage showed a soil carbon content the plus 166 kilogram CO2 per hectare per year. In contrast, conventional treatment minus 4,000 kilogram CO2 per hectare per year. So tillage treatment is very important. So the first half is that I show the importance of agricultural practice in relation to the global warming CO2 emission. Second half, I talk about nitrogen, but the nitrogen metal is very complex. I made the it's a chemical lecture. Nitrogen is oxidation number +5 from +5 to -3. I put each compound nitrate, ammonium, plot here. nitrogen gas is the zero. Yes, profile here. And microbes involved in the conversion of these compounds, like it's very complex. Famous one is the nitrification, is the ammonium to nitrate. But the quite complex. Denitrification is a reverse way from nitrate to nitrogen. It's a main route, but the many kinds of microbes involved. So N2O, nitrous oxide, is a strong global warming potential gas. It's quite many organisms can participate in this gas formation. That is a very complex phenomenon occurs in the Earth. So we control N2 emission. In that case, we control these kind of microbes. It's difficult. So there are many researchers, the role of each microbe in the in formation N2O. So I showed rapidly, yes. One important thing is the water content. If water content increase, in that case, substrate diffusion increase, but oxygen diffusion limit. So this is a very important scheme. This is important for thinking of the N2O. Denaturation is another process, so the water content increases, the denaturation occurs much higher. But in contrast, the nitrification is lower. It is higher. That the limit oxygen diffusion, so the nitrification occurs. So the product is mainly N2, but intermediate water content, in that case, N2 appeared. So soil water content influence the N2 emission content. Burma, it's a quite old comment. The soil water content and the timing of fertilization had far greater than fertilizer composition. High rate of application of nitrogen fertilizer common in the developing, mainly tropical cultures are likely to increase. Excess for rice cultivation, most aggressive production in tropics on the well-drained soil. So the condition of high rate of nitrogen fertilizer use and the anaerobic state largely unrestricted diffusion in soil concern favor large gas fluxes. Tropical agriculture land is potentially large emitter of N2O and N2. Yeah, I explained here the effect of O2, the N2 production. Also, the pH affects the N2 production like this. And differently than nitrification or ammonium oxidation, other nitrification. So it's a very complex way. pH of oxygen influences the activity. And also the many kinds of the factor involved in the N2 emission. In this case, the nitrate concentration low to high, and nitrate substrate, organic, ammonia, and nitrate. So these organisms have a role differently the activity. In this case, heterotonitrophile is mainly low nitrate concentration. Co-denitrification occurs in the presence of high nitrite content, like this. So N2O emission partly depends on nitrite concentration and also the depending on the substrate difference. I also show the recent progress with the genetic approach. Now we understand the nitrification process is different, involved by different enzyme. Just the NAR, NAP, NIR, NOR, NOS. The NOS genes are classified two clade, one clade, two. These results show the complete reduction from NO3 to N2 completely one can perform, and non-derived N2 reduction. Some soil microbes does not complete, but have a role in this process, and sometimes here. So we believe or we understand the nitrification is complete by every denatured microbe, but recently we understand incomplete or non-denaturing N2 reduction occurs. And in soil, recently, this type much abundant in the soil. So, non-denaturing N2 reducer, gene named created to NOSZ, probably this microbes important role consumption N2O in soil. So recent researchers are trying to the N2 this type of organism directly put into the soil and to prevent N2O emission from soil, and they got the positive result. Still the discussion needed, but their data shows 11 different soil and effective N2O reduction in the 100, almost 200% in the case of 1/3 soils. So still that we need the study, but this kind of microbe use is very important for the further control in the N2O emission. So I right here, I mentioned about old carbon use and fresh carbon use. And old carbon use is mainly industrial activity. And the other hand, the fresh carbon soil, the agricultural activity, it's a bi-direction, yes. But it's a one way. So agricultural plastic is very important for the maintaining of the soil carbon. Also the climate change and also affect crop yields, positive or negative. Yes, also the biochemical cycling, C and A, any change that is on the agricultural practice. Yes. My conclusion was that this lecture here, microbes are the key decomposers of organic matter and release nutrients in the soil for plant growth, as well as CO2, methane, and N2O into the atmosphere. Agriculture activity combined with the microenvironment factors, including soil type and water content, greatly influence the complex interactions. These interactions dictate how microbes respond to and affect climate change, and how climate change in turn affects microbe responses. And lastly, I show the photo. Do you know what is this? It's a dead fungal body. The green color, small cell. Now yeah, at that time, eat fungal body. It's the microbial carbon, quite labile organic carbon. Easily CO2 emitted from here. And humified materials, it's very difficult to decompose. Yeah. So always I imagine this picture. Microbes is make some action in the soil, so that we need to imagine soil microbes for the also that's a link to the global environment. So my university here, University Library.
Okay, thank you very much, Dr. for your insightful lecture. Now we are going to have a discussion. I would like to invite the students. Any discussion? Any question? Please raise your hand. Yeah. Yeah, you have. Please come forward. Yeah. Thank you, sir, for the question that you asked. Let me introduce myself first. My name is Yana Kerusa.
Yeah.
I'm third year student
Yeah.
in here and then so, I read one of your related study journal, especially about Indonesian farmers can contribute in reducing greenhouse gases emission from wetland life cycle, right? It's one of your study journal.
Yeah.
And as we all know, the greenhouse gases mainly consist CO2, CH4, and others. In rice-producing countries, such as in Indonesia, farmers can contribute significantly in reducing greenhouse gases, right? But why do you think this could happen in Indonesia? Because I know Indonesia not implemented this reducing greenhouse gases. And for you, why do you think this could happen? Or are external factors causing it not applied in Indonesia? Thank you, sir.
Indonesia is a tropical country, so emission from gases from soil is, I suppose, it's quite high. So, and also the Indonesian agriculture is very important for control CO2 emission. So, our scientists need to discuss think about the environment. CO2 and energy emission. So the net is very tough. Then, always we need to discuss about scientific and our so on. So, and yes, Indonesia have important point in the social problem in Indonesia. Yes.
Yeah, in Indonesia, the still many village, mostly the agriculture sector still cultivate using village. We are not familiar with the village.
And about the technology, we tried the biofuel project. That is a biofuel. Sugarcane is popular in the tropical country, and amended to the making ethanol is used in place of the petroleum. That is at that time that is a good way, but it's the cost of making biofuels. At that time, the cost is not so cheap comparing with the petrol. But now, in this, especially this year, energy problem occurred. So one possible way is the improvement of biofuel production. Yes, and the Indonesia is a suitable country for such production. It's the carbon neutral cycle. This is one way. And I also imagine the solar panel construction, the independent carbon. But our side is agriculture, keeping the food production. So we need to keep food security and energy security. So the agriculture production is still in. So it's the much discussion needed for. That's it okay?
It's okay, yeah. Further discussion. Okay, one more question because the time. Okay, I want to invite one question. Any?
Yeah. You.
No question anymore?
I hope you in
Microbiology.
yeah, good. Yes.
Microbiology and ecology.
Yeah. I suppose your study is very important for the sustainable future. So now I mentioned the biofuels, also the lastly, as I showed the good microbes, we discover good microbes. Depress activities, electron emission and CO2 emission. That was a good source for the make good environment conditions. Your study is very important. Please study much harder.
It's very hard. Oh, is there any question from YouTube? Please help me to look up. No question? No question from YouTube? Because I think the time is over. Thank you very much. Thank you very much.
Thank you for your attention.
Yeah, say thank you very much also to you, our student, and my dear college lecturer for joining lecture. Hopefully you can get new insight, and we can come back with the discussion. Okay, thank you very much, and in the examination. You send the material. Later can you send your PPT? My email.
Yeah, okay.
Can I distribute to the student?
Yeah,
Is it okay?
okay.
Later is okay.
Okay. I